Hello, and welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the second episode of Plantopia for 2025, and today I welcome Dr. Marisol Quintanilla, assistant professor of nematology in the Department of Entomology at Michigan State University in East Lansing, Michigan. Dr. Quintanilla completed her Bachelor's of Agriculture from Andrews University in Michigan in 2001, followed by an MS and PhD in entomology from Michigan State University. Following completion of her degrees, she served several roles, starting with program leader of Northern Marianas College Cooperative Research Extension and Education Service from 2009 to 2011, junior researcher with the University of Hawaii Plant and Environmental Protection Services, extension agent with the University of Hawaii, and applied nematologist with her current department from 2017 through 2019. Dr. Quintanilla began her current role as assistant professor in 2019. Her research and extension work focuses on plant parasitic nematode management as a component of sustainable soil health programs in horticulture and field crop production systems. Her work seeks to find applied solutions to plant parasitic nematodes in vegetables, fruit, and ornamental crops in Michigan and beyond. Dr. Quintanilla is an active member of APS and the Society of Nematologists, and is the recipient of the Society of Nematologists 2023 Syngenta Crop Protection Award for early career scientists who have made significant contributions to the advancement of nematology and agriculture. Marisol, it's great having you here on Plantopia.
My pleasure, Matt.
So you're currently an assistant professor of nematology at Michigan State University.
It's the Department of Entomology. I'm the only full-time faculty in nematologist.
And I bring that up because there's a lack of expertise in nematology across a lot of the land grant systems. There might be a single nematologist that's at an associated research center off campus, and therefore the students don't have the option to take nematology.
And that is exactly right. There's a lot of universities that don't have a nematologist. Mm-hmm. Some have one. There's only, to the best of my knowledge, 2 universities that have a nematology department. The rest of nematologists are inside entomology departments or inside plant pathology department.
Now, a number of listeners may be familiar with plant diseases and some of the causal agents, especially if they've listened to previous episodes, but maybe not all our listeners know what nematodes are.
Nematodes are microscopic roundworms. It's a very big animal group. There's nematodes that feed on plants, and those are called plant parasites. There's nematodes that parasitize insects. They're called entomopathogenic nematodes. There's nematodes that are parasitic of humans. There's nematodes that parasite animals, like dog heartworm. There's nematodes that feed on bacteria. There's nematodes that feed on fungi. Nematodes are a lot like insects. They fit every trophic group.
Right. If you were looking at nematodes across different trophic levels, how would you tell a plant pathogenic nematode from a non-plant pathogenic nematode?
Well, plant parasitic nematodes or plant pathogenic nematodes need to feed on plants, and plants have a pretty strong cell wall. Mm-hmm. So the plant parasitic nematodes need to have a strong stylet. It's like a hypodermic needle that can pierce the plant cell wall and then feed on the plant contents. Mm-hmm. While fungal feeding nematodes can be very similar and they also have a stylet, it'll be very fine and very dainty because fungi in general are pretty soft. They don't need a strong stylet. Bacterial feeding nematodes don't have a stylet. They have just like an opening in their mouth. They swallow the bacteria. So plant parasitic nematodes have different anatomy that fits feeding on plants.
They're similar to Hemiptera in that way, that they have piercing, sucking mouth parts.
Yes, exactly, exactly. Very similar to Hemiptera. In fact, some of the plant defenses that works against things like aphids also have an impact on nematodes.
Now, nematology is underappreciated compared to some of the other subspecialties in plant pathology because, like a lot of soil-borne pathogens, a lot's happening beneath your feet. Is that challenging to get people to understand and appreciate nematodes because you have to rely on these specialized techniques to see them?
Exactly. Most people and most growers do not think about them. So it's like out of sight, out of mind. Because most plant parasitic nematodes are soil dwelling, they are really invisible and they're microscopic. So most people don't know that they're causing the damage. And many times the damage is attributed to nutrient problems or some other pathogens. People many times don't connect the dots because those symptoms are in the roots. Like for root knot nematode, you have the gall like knotting in the roots. For cyst nematode, you can actually see the cysts that look like little grains of sand in the roots with your naked eye, but it's very small. But most other nematodes, the damage is it's only can be assessed by sending a sample to a lab in which they extract the nematode from the soil and look at it in the microscope.
Right. And the other thing that I think the listeners will appreciate and understand why it can be challenging in diagnostics and management is that nematodes, like a lot of soil-borne pathogens, are ever present. It's just that their abundances change. So if someone sends in a sample, the likelihood you'll find some plant pathogenic nematodes is really high. But the likelihood that the populations will be high enough to cause yield losses depends on what's planted there and how it's managed, right?
Yeah. To give you an example, Matt, my lab has done several nematode surveys in the state of Michigan. And, vegetable, carrots, soybean, corn, I could keep on going. But we have done survey in which we sample different farms. In all of our surveys from all of our crops, more than 90% of samples, and some crops, more than 95% of samples have root lesion nematode in them.
Mm.
So root lesion nematode in Michigan is something that is widespread. I think it's easy to assume that everybody has it, that everybody has root lesion nematode. There's several species of root lesion nematode in Michigan, but root lesion nematode, almost everybody has it. Soybean cyst nematode, almost everybody that grows soybean, but in every county that grows soybean, it has soybean cyst nematode, either recorded or not recorded yet, but is widespread amongst soybean growers. Root knot nematode, northern root knot nematode, Melodagaina hapla, 56% of our samples have northern root knot nematode. So we're talking about they're widely distributed, very common. Of what makes a difference is sometimes their numbers are above damage threshold and sometimes their numbers are below damage threshold. So the plants can withstand quite a bit of damage. Like, for example, it's not uncommon to have 200 individual nematodes for 1 cubic centimeter of soil. So that's a small cup of soil. So you're talking about a lot of them. And in a root, also, you can have hundreds of them in a gram of root. So we're talking about they're very common, very widespread, but plants can tolerate quite a few of them before the damage threshold is reached. The problem also is that nematodes often make the plant more susceptible to other pathogens, and some even actively transmit pathogens. Like some dagger nematodes can transmit some tomato ring spot viruses, some viruses. Root lesion nematode can make potato more susceptible to Vericillium dahliae, which causes potato early die. And many nematodes, there's association between a nematode and another plant pathogen. And many times that plant pathogens have much more devastating consequences on the plant than the nematode itself.
Yeah. Well, that's a perfect transition and talking about it's something important that you've worked on that I think is also insightful for listeners to understand is that there's a difference between a disease and a disease complex. And you work on nematodes, and a lot of nematodes, like you said, form partnerships with fungi, with viruses, with bacteria. And oftentimes we give the credit to the virus. We'll say, oh, it's tomato ring spot virus. But, well, yeah, it needed a vector or needed predisposition of the tissue, and I think in that case, it's not giving nematodes credit where credit is due, because if there's not that change in the chemistry of the plant tissue, you can't get subsequent invasion. Or if they're a vector, it requires that specific nematode to be able to transmit that virus.
Exactly. So in the case of tomato ring spot virus and dagger nematode, it is a required vector. It's a lot like mosquito and malaria or mosquito and dengue.
Right.
You guys all heard of mosquito and dengue or Zika virus. People don't get dengue, people don't get malaria by being friends with somebody else or eating from their food. They get it from a mosquito feeds on an infected person and then feeds on a healthy person. The mosquito acts as a bridge, as a vector. Vector means bridge. So nematodes can be vectors of certain diseases, especially viral diseases. And dagger nematode is famous for that. So pretty much for plants to get some of these viral diseases, you can get it through nematode. Like, for example, if you have pruning hooks and you prune the plant that's infected and then you prune a plant that's healthy, you can also transmit it that way by having, but it is not, it's something that the nematode actively vectors. There is some, like for example, root lesion nematode and Verticillium dahliae is not that way. It's more of the nematode carries the infectious organism, mainly like accidental, and also Verticillium dahliae is something that if the nematode is not there, the plant can also get Verticillium dahliae. They can also get the fungal infection. Just the nematode makes the plant more susceptible to it. And that's a fungal disease. So in cases of nematodes, nematodes can be vectors of viral pathogens, but most of the fungal associations, it's not considered a vector. They're not considered vectors.
Right. It's not stylet borne.
Yeah.
And what you're getting at is that they provide infection courts for subsequent infections, but they also can interfere with plant defense responses.
That's right.
And I want to get back there because we kind of open with that, and we were making this comparison between aphids, for example, and dagger nematodes or something like that, where you have these piercing, sucking mouth parts. One's above ground, one's below ground. But we also know that there could be localized responses by the plants in terms of defense, maybe a hypersensitive response, programmed cell death or something.
Yep.
And then there could be the systemic acquired resistance in plants. And we know that there's different pathways depending on if a plant is experiencing herbivory or attack by insects versus if they're being invaded by fungal pathogens. Are you saying that, like aphids, that when nematodes invade plant tissues, the plant response is similar to as if it's being invaded or attacked or fed upon by insects?
Well, there's different kinds of nematodes and their feeding is different and the plant reacts differently. For root lesion nematode, they're migratory. So a lot of the damage, they're moving and they're moving throughout the cell and feeding in different parts. While root knot nematode and cyst nematode, they're sedentary, they find the place, and they actually hijack the plant systems. So they communicate with the plant chemically, and the plant provides it a special cells, sometimes multinucleated cells that are hyperfed, and the nematode doesn't have to go anywhere for the rest of its life. It's getting food supplied by the plant. So the plant starts giving priority to feed the nematode. So not all nematodes react with the plant in similar ways.
Right.
But yes, the immune responses of the plant are affected in different ways depending on which nematode and which damage.
Sure. Yeah, that brings up some interesting points. And one of them is maybe our listeners have heard more content related to fungal pathogens. And there's a lot of fungi that kill tissue and then are able to absorb those simple sugars as a result of breaking down complex substrates. Others are biotrophs like rust pathogens, for example. They have to feed on living tissue. And like these root knot and cyst nematodes are more like that because they have to stay in place till they can fulfill their reproductive cycle. And
Yes.
they can't do that if they kill their host too fast. So they have to keep feeding on it so they can feed their young.
Yes. In fact, almost no nematode kills their host.
Mhm.
So nematodes in general don't kill their plant host. They will reduce yield,
Mhm.
reduce growth, but not kill it. I think most of plant parasitic nematodes, they are not benefited by the death of its host.
Mhm. So we've talked about the different styles of feeding of nematodes, and a lot of your work on plant parasitic nematodes are on, basically soil dwelling nematodes. They're feeding on the roots. But clearly there's foliar nematodes, and one that's gotten attention in recent years is this one that's causal agent behind beech leaf disease,
Yeah.
which is a really problem. It's a big problem on beech throughout a number of states in the Northeast and in the Mid-Atlantic. Do you think that the emergence of beech leaf disease has been useful for trying to justify why nematology is important? I mean, certainly like soybean growers and other things that deal with these kinds of cyst nematodes and problems. But I think the general public doesn't really understand nematodes. But when you see something like beech leaf disease, which is foliar, and just right in front of your face, you're watching these trees lose their foliage, die back. Not that we're celebrating the disease at all, but do you think it's been useful in helping to engage with the general public on why nematodes are important and what they can do if they're, introduced?
I think for the general public, yes, this has brought publicity to nematodes, which are not very famous. They're just not very famous because at least plant parasitic nematodes mostly are in the soil and the roots, and most people don't even know they exist. For the kind of people I interact with, I work with growers and ag professionals, and most of the growers are familiar with nematodes. I have been working with them for many years. Soybean growers don't need anybody to tell them about soybean cyst nematode. For soybean, it's the pathogen that takes the biggest cut or the biggest yield cut in soybeans. So they are very aware of this. Most growers have heard of nematodes in some way or another. In the past, no. The reality is that nematodes have quite an impact on yield and plant growth. So farmers are beginning to know about it, especially the ones that go to extension meetings and go to trainings. Those are the ones that I interact with the most. The ones that don't come to my talks, I don't interact with them. So the ones that I interact with mostly know about nematodes. Among the general public, I would say beech leaf disease has really increased awareness of nematodes. I would agree with you on that. It's very drastic what's happening to beech trees. Unfortunately, it's not something that will be easily solved. There are some effective products that have been evaluated. There have been some good papers with some good products that work, like injection of trees. But that's something that's going to be used maybe for a few trees in a campus, in a park, but not in a forest, where you have thousands of trees. That's not going to be economically viable.
Right.
We're just gonna lose a lot of beech trees.
So I want to pivot back to talking about yield losses. Clearly, you work on cyst nematodes and root knot nematodes, beet cyst, the hop cyst. a number of important cyst nematodes on important crops. What kind of yield losses, like which of these that you work with actively, like let's take soybean cyst nematode. If it's undermanaged or not managed, what kind of yield losses do we see on average across a given season? Are we talking 50% yield loss or more? I mean,
Before we had soybean cyst nematode resistant varieties,
mhm.
because there was a time, soybean cyst nematode has been recorded first in the United States in the 1950s, but people were not really aware of soybean cyst nematode, or was not widely distributed until like the 1980s. And there was no resistant varieties. And there came a time where yields were, you could say, 100% because it was not worth it to harvest.
Okay.
It was, you would lose more money harvesting than not harvesting because there was such little yield. So if you have susceptible varieties and you have high populations of soybean cyst nematode, it is absolutely devastating. That led to looking for resistance. There was trips to China, Asia, Japan, looking for resistant varieties. One very effective resistant variety was PI 88788. PI stands for plant introduction. So plant introduction 88788. It was a game changer.
Okay.
You could still have excellent yields in the presence of soybean cyst nematode. This has been grown for 25 years, maybe, consistently, PI 878. More than 90% of soybean varieties grown in the north of the United States, in Michigan, have PI 878 source of resistance. A concern is that nematodes are breaking the resistance. They're getting adapted and they're beginning to be able to reproduce in this. So now we're getting 5% yield loss, 10% yield loss, depending of how the numbers and how resistant. There's different populations. There's places where the resistance has broken down more than in others. Farmers that rotate with non-host quite a bit are doing better than farmers that don't. And just growing corn and soybeans alternatively, it's not enough. When I did a soybean survey, the growers that rotated with a third crop, let's say corn, soybean, and wheat, or alfalfa, or anything else, they did much better than just corn and soybean. So there is yield losses, but it's not catastrophic as before, mainly because of resistance. But that resistance is breaking down. Therefore, I recommend growers to rotate sources of resistance. Unfortunately, there's only two choices available commercially, PI 8788 and Peking. I don't recommend growers to switch to Peking because in my trials, confirmed by others, Peking resistant breaks down faster than PI 8788 resistance. So if growers only grow Peking, their yields fall faster.
Right, right.
So they need to rotate.
Well, the question that arises then is when they rotate to corn or they rotate to something else, I assume they're not getting the return on investment. Like of those three, you get the most financial payback from growing soybean, right? Okay, so there's a disincentive to rotate away from it. Even if you have a 5 or 10% yield loss, you still might profit more than if you rotated to corn. And I think that's a short-sighted viewpoint to take. But if you're putting yourself in the perspective or in the shoes of the farmers that are, working the land, they might say, well, it's worth taking a 5% loss, not realizing that it's going to have a devastating effect if it breaks down even more. And then that 10% is now 25%.
That's right. That yield loss increases every year. And fortunately, PI 8788 has been an excellent source of resistance that has held up for so many years. Like I said, like 25 years, it has held up. And the yield losses are decreasing pretty slowly compared to Peking. In Peking, the yield losses is much faster. But it is happening. And the problem is that we don't have excellent alternatives to P8788. There are companies who are researching this. There are researchers in university like Melissa Mitchum in the University of Georgia, who is researching this, and she has found excellent alternatives that, to the best of my knowledge, are not commercially available yet. So there are things in the future. There are things in the horizon. But it's important to protect what you have. And there are other crops that are profitable, It's very difficult to have specialized machinery for some of these crops, but they can rent to somebody else who specializes on another crop.
Coming back to the growers, obviously part of your appointment is extension. You spend a lot of time with growers communicating these results and talking about breakdown and resistant varieties and the importance of trying to slow that further. Are the growers responsive to those suggestions? I know that's kind of built on like a legacy of trust between university and the larger community, but do you feel like in your area and in your specialty and in the crops you work in, you have the trust of the growers?
Yeah. I mean, I am part of something called the SCN Coalition. Soybean Cyst Nematode Coalition is a pretty big group. It has a huge impact. Millions of people have been impacted, seen their videos, seen their material. There has been huge change in behavior. In fact, it was the original SCN coalition that introduced the use of PID 788, which pretty much solved the problem. So it is now the new SCN coalition who is sounding the alarm that this wonderful tool is breaking down. Many farmers understand this. In a recent survey that we did, about 20% of farmers are now rotating picking with PID 788. I wish it was higher, but at the same time, it's not easy. Not all seed companies have picking alternatives, and growers, they have like their seed salesmen, and it's not easy for them to introduce a picking in their rotation. And also, there's some downsides. If they put picking in the rotation too many times, then picking will also not work very well. I am telling farmers that I prefer them. My recommendation is that they grow 1 year of PID 788 soybean, 1 year of a non-host, and then the next year again PID 788 and then picking. So do 2 PID 788 and then 1 picking. So alternate it in such a way that picking is on the third time, in part because you want to save picking, and every time they grow picking, they will get a yield bump because they have a different source of resistance. So they'll get a yield bump and they'll be excited, and then maybe they'll want to switch to picking, but then they'll have declines after that. To keep the yield effectiveness, you need to not grow it every year.
Sure. Well, we've talked a little bit about crop rotation, crop diversification. You're talking about cultivar rotation and scheduling of that. In addition to resistant varieties, especially with this breakdown of resistance, are there other strategies besides the crop rotation? Clearly, a lot of the historic fumigants we've used in soils, and maybe that's more of a greenhouse kind of thing, they've been kind of taken away as an option. But what other options do we have for controlling nematodes when we can't rely exclusively on resistant varieties or crop rotation?
Yeah, well, fuming, methyl bromide is very rarely used anymore, except some very specialized crops. It worked very well, but there was some environmental concerns, the effect on the ozone layer. There's Telone, Vapam, but some of this, like for example, Telone, is used on things like potatoes, strawberries. They're not used in soybeans. The cost per acre is not justified for some field crops.
Right. Because the return on investment isn't high enough.
That's right. It's about the return on investment. For some crops, it is justified to do fumigation. For some crops, it isn't really. What other alternatives that can be used? Rotation with non-host is one of the most effective methods. Another thing, there's some cover crops that really have a special power to reduce nematodes. Like there's some varieties of oilseed radish that can be very effective to reduce some nematodes. If you're talking about northern root knot nematode, all grasses are a non-host. I've also had quite a few trials with compost and manures and have found that compost and manures that are based on chicken or poultry manure or swine manure, but especially chicken and poultry manure, I found them to be quite effective at reducing nematodes. In some of my trials, if you have a 5% concentration of compost and manure and 95% soil, you get complete mortality with chicken manure and some other products like layer ash plant from Morgan Composting. But in practical for farmers, that would be immense amounts. So in amounts such as 2 tons per acre, 1.25 tons per acre, which is commonly what growers apply, there is a reduction of nematodes, but not an elimination. But they do help in the management. There's also some nematicides that can be more or less effective. Unfortunately, none of them are extremely effective, especially for soybean cyst nematode.
Is there a lot of new nematicides coming on market? I mean, are there a lot of companies actively working on developing them, or is it there's just a lot more attention on the breeding side? Breeding for resistance seems to be the most effective strategy.
Well, in the last few years, there's been quite a few nematicides that have come in the market that are way more specialized on the nematodes and don't have broad spectrum.
Right.
Like fumigants or like Vite, also very broad spectrum, affect a lot of organisms. A lot of these nematicides are more specialized in nematodes, and some of them can be quite effective. The problem is with soybean is that most of them are it's on the seed coat, like a seed treatment. And I think the amount of the product is not enough to protect the plant on the long term. In most of our trials, we'll see a protection for the first few weeks, but then the product is gone. The product protects the seed,
Right.
and the first few roots, but after the plant grows up, there's not enough of the product, or there's none of the product left over to really protect the plant. So the nematodes can increase after that.
Okay. I don't think it's a hard sell to tell people that something that impacts soybean is going to have a devastating impact on American agriculture because soybeans are such an important crop in the United States.
Yeah, for sure. In some states, you can drive for miles and all you see is soybean and corn.
And it makes sense that there's active breeding programs and development of nematicides because there's such a good return on investment. They could afford to invest dollars to improve cultivars and varieties. Now, you also work on cyst nematodes and have published work on cyst nematodes that are on maybe less desirous crops in some ways. People will say soybeans, hops, I mean, like beer, but like let's take beet cyst nematode, right? Like I don't know, or the beet cyst eelworm as it's called. What's the deal with that? And like how much in terms of what's beet production look like in the United States, and how is something like beet cyst nematode impacting its production, and where is it impacting its production?
Yeah, well, beet is the sugar beet. So most of sugar production in the United States is from sugar beets. When you buy sugar, most of the sugar that you buy is not from cane sugar. It comes from sugar beets. Sugar beets are very similar to the red beets, but they're white, and mainly the plant juice is extracted and then the sugar is dehydrated, etc. But so mainly is for sugar production. And yes, there is quite a bit of sugar production in the United States. In several states, Michigan has in the thumb, Michigan looks like a glove. In the thumb area, there's a lot of sugar beet production. And it's produced by Michigan Sugar. Michigan Sugar pretty much coordinates all of the sugar production in Michigan. And there's other states that also produce sugar. Sugar beet cyst nematode is extremely similar to soybean cyst nematode. It is so similar that they're very hard to tell apart, and they can breed. So they can breed and produce viable offspring. Their main difference is that, beet cyst nematode prefers to feed on beets, and the soybean cyst nematode prefers to feed on soybean. But other than that, there are not too many other differences. Their biology They're very similar in their behavior. And beet cyst nematode reduces the yield and the quality of the plant, and it can have quite devastating effects. It can reduce the yield and the sugar yield significantly. And it's a very important priority for sugar beets. Some, in fact, breeding companies in Germany is a place where beets are commonly grown. And I think it's maybe where sugar beet cyst nematode might have come from. But varieties have been developed that are tolerant or resistant to sugar beet cyst nematode. And also in the United States, a lot of sugar beet varieties are sugar beet cyst nematode tolerant, so that they can still yield well in the presence of sugar beet cyst nematode. There are also resistant varieties where the nematode can't reproduce well in the sugar beet, but they tend to produce smaller beets. So you need to have, higher seeding. And in the United States, usually farmers prefer to grow tolerant varieties and not resistant varieties.
Okay. Now, hop cyst obviously impacts hops. I know on the West Coast, there's a lot of hop production in the Northwest. Is there a lot of hops grown in Michigan? And what's the U.S. hops market, and how is hop cyst impacting hop production around the United States? Or is it more a local issue?
Hop cyst nematodes is a devastating problem. It can reduce the plant growth by 40-50%. We have done some trials, in which we'll have plants, hop plants with hop cyst nematode and without hop cyst nematode, and recorded their growth. And the plants with hop cyst nematode will be somewhere between 240 to 45, 50 smaller and produce less cones. So for sure, hop cyst nematode is a severe problem. Hop cyst nematode can be found in the Pacific Northwest and Washington State. We have done surveys. My graduate student, Elizabeth, has been involved in sampling in Washington State and in Michigan and done surveys. And unfortunately, hop cyst nematode is found both in Michigan and in the Pacific Northwest. It is a devastating nematode that is mainly being moved around with planting material. When they grow the rhizomes, many of these rhizomes already have hop cyst nematode with it. So that's how it's getting around.
Has that led to quarantines and things like that? Or is it they're still trying to delineate where it is? I mean, how new is it?
Well, for Michigan, I think that the first report was pretty recently. It was the first finding was by Fred Warner and Dr. Bird, I would say maybe 15 years ago. I don't remember exactly, but it was only one farm in which they detected it. We've actually had done a survey and did several farms, and it's pretty commonly distributed. A lot of farmers have hop cyst nematode, and also it is found commonly in Washington State. There's not a lot of quarantine. A lot of the plant, the clean planting material will be cleaned for viruses. They check it for viruses and not necessarily for hop cyst nematode. And we think that hop cyst nematode, because of the tremendous impact, reducing the plant growth 40 to 50%, should be a priority in providing clean material.
One of the things that I can't help but think about when I think about nematodes is challenges in doing molecular work when you have very few individuals. I mean, yeah, if you have an infested soil, or you can do extractions from a stunted root system that has just tons of nematodes, and you can get material. But I feel like in some ways there's been this over-reliance on the traditional morphological characters in nematology. This is true of some of the subfields of entomology too. And do you think that nematology has caught up and kind of came to meet the challenges of the molecular age with molecular diagnostics for nematodes, or do we rely disproportionately on morphological ID and things like that?
Well, I personally work mostly morphologically, But I do have students that have focused on molecular techniques. A lot of the species identification is done with one individual nematode.
Mhm.
So you can do molecular identification from one individual nematode. And that's the way most of our work is done, with one individual
Right.
nematode. So nematology as a field, I would say it's always a bit behind, because we're always a bit behind plant pathology and entomology. I think in part because there's less of us. So we're always a bit behind. At least for me, I collaborate with other people. So there is a faculty member in my department who helps me with the molecular work. He's an excellent molecular scientist for Drosophila, a fly. And we use his molecular skills and equipment to do our work. And that way we can kind of stay up to date. So that's the way we kind of stay up to date, is with collaborating with people who are using molecular techniques every day.
Right. And I feel like, having taken nematology myself when I was at Penn State and understanding the difficulties in getting to get the pattern just right, to make a slide, to identify things, it takes some highly specialized skill to make those, and even to pick up a single nematode if you're not
Maybe
used to
that's
it.
the hardest part.
It is challenging. So I think that there's a real need for molecular tools and molecular diagnostics in nematology, because there's not enough people with your expertise to be able to look at something. I mean, usually they're heat fixed, but I mean, something could wiggle by on a microscope slide and you can identify it, even if it's moving fast, because
Yes,
you,
yes,
you
yes.
recognize its features. But there's not enough training in plant pathology programs across the US, where I think there's a real need for molecular diagnostics. And like you said, there's people that can do that, but I think there needs to be support for that kind of work, because diagnosticians that work in labs and they wear a lot of hats. They have to be able to recognize these things. And by process of elimination, they could be like, well, it wasn't a fungus, it wasn't a bacterium, it wasn't this. It's probably a nematode. Okay, we did a soil extraction. We see plant parasitic nematodes. We can look at the stylet and identify some features. We think it might be this genus, but having those molecular tools, I think, are really important, especially when we get
For
down to,
sure.
yeah.
It's going to move more and more in that direction, because you can get higher precision with less specialized skill, because it's a steep learning curve to become a nematologist by identifying things with morphological characteristics. It takes quite a bit of time to get
Right.
there.
Right. Well, we're actually just about out of time here. I definitely enjoyed talking with you today. It's nice to have researchers like yourself who are not only doing extension, but are working on plant pathogens and plant parasites that I think are underrepresented, but still represent a huge amount of biomass in our soils.
My pleasure, Matt.
We've just heard from Dr. Marisol Quintanilla, Assistant Professor of Nematology in the Department of Entomology at Michigan State University. Be sure to check out the show notes section immediately following this episode description for links to Dr. Quintanilla's research. I'm Matt Kasson, host of Plantopia. Thanks for listening.